Welcome to The Ultimate Asset, the podcast where elite physicians, health strategists, and high-performing leaders explore what it really takes to protect and elevate your most valuable assets, your body and your mind. This is high-performance medicine, powered by Velocity Health. Hello, and welcome back to another episode of The Ultimate Asset. My name is Dr. Deep. I am your host, physician, practicing precision medicine physician. As you know, we often bring themed months to you, talking about various topics. This month, we're thrilled to be chatting about something that's very near and dear to all of our hearts, which is cardiovascular disease. As some of you have heard of me talk before, I have three grandparents who have passed away from heart attacks in their 60s. My father's had multiple stents, a first cousin with a quadruple bypass at age 45. This is something that's real to me. Number one killer in the world, obviously. We are thrilled today to be bringing on a guest who will be educating us on this topic, and we'll talk about all sorts of interesting aspects of cardiovascular disease. Dr. Stephen Nissen. Dr. Nissen is one of the most prolific physicians and researchers in the world. Frankly, we are extremely excited to be chatting with you. He currently serves as the Chief Academic Officer of the Heart and Vascular Institute at the Cleveland Clinic. Started your career, I believe, at the University of Michigan, UC Davis, University of Kentucky, and then joined the Cleveland Clinic, and has been a storied researcher, trialist. We'll talk about some of his landmark trials. But Dr. Nissen, thank you so much for joining us. Welcome.
Well, it's a pleasure to be with you. We'll have an interesting conversation.
Yes, we will. Dr. Nissen, I want to start with potentially something that most podcasters don't ask you about, which is, tell me a little bit about Dr. Nissen, the man. Where did you grow up? Did you know you were going to be a physician? How did you sort of come to cardiology and the work that you're doing right now?
Well, I grew up in Southern California. My father was a physician, OBGYN. And, it took me a while to figure out that I wanted to become a physician. But I made that decision while in a very long time as an undergraduate at University of Michigan, longer than most, because I was very much involved in the anti-Vietnam War and civil rights movement, in the 60s. And so that kind of kept me occupied. But then I pivoted and went to medical school, went off and did, my internship and residency in internal medicine at UC Davis. And I met a legendary guy, Dr. Tony DeMaria, who was kind of a mentor to me in the early phase of my career. And he, during that early phase, really helped me understand how to do research. And he went on and became the editor of the Journal of the American College of Cardiology many years later. And I went off to the Cleveland Clinic now 30 plus years ago. And that's where I've been ever since.
Amazing. Well, thank you for sharing that. You've led some of the hallmark trials that in all of medicine, frankly, and it's interesting to hear about your sort of advocacy in the Vietnam War. I wonder if some of that has played out in your patient advocacy for patient safety as you've researched and exposed, frankly, various harms that are in our healthcare system. I want to talk about three broad sort of categories today. One is your prior work. We'll talk about some of the trials that particularly hit our work as we chat about longevity and precision medicine with many of our patients. We have a high IQ, high sophisticated audience who's really interested in this kind of work. Second will be lipoprotein A. And lastly, we'll just kind of finish off with directions forward. So, Dr. Nissen, we just had Men's Health Month here on the Ultimate Podcast. We were talking a little bit about testosterone replacement therapy. I just want to spend one moment here and then focus the rest of the conversation on more cardiovascular issues. But you did lead the Traverse trial, which was a landmark trial looking at cardiovascular outcomes in men on testosterone replacement therapy. Something we talk about a lot. We get a lot of questions about from our patients. We have a very thoughtful and strict approach to when we will start folks on that. But one of the myths that I think this trial debunked was that testosterone replacement therapy can increase cardiovascular outcomes. It'd be great maybe for you to describe in your own words, what did that trial actually tell us and what did it not tell us? And how do you think we should use those outcomes in our practice?
Well, that's obviously a very important question for men as they age. First of all, please keep in mind that testosterone levels do decline with age. It's not necessarily a disease. It's like what happens with women when they go through menopause. I actually call it menopause.
Yep.
But there are men who develop symptoms, and they can have fatigue and low libido, worse sexual performance. And this therapy that began to be used very commonly. FDA had a lot of concerns, and they told the 11 companies that make testosterone products that they were concerned about cardiovascular safety and they needed to do a cardiovascular outcome trial. The lead was AbbVie, because they made the most commonly used product, which is a gel containing testosterone. And AbbVie came to us. I have a team here of about 100 people that help me do randomized clinical trials. And I think it came to us in part because, we've had a history, I've had a history of when there are harms of, being very open in publishing those findings. And so that provided some credibility for them. The trial was very difficult to do, because the men that entered the trial had high expectations. When men take testosterone therapy, they think it's going to make them feel like they were 25 years old again, and that just isn't going to happen. But we did this. The trial was done carefully, where it was randomized to either testosterone gel or a matching placebo. And then the dosing was titrated to get men into a normal range for testosterone. If they went too high, the dose was reduced. If they weren't high enough, the dose was increased. They were seen very frequently and then followed for cardiovascular outcomes. Now, I want to make sure everybody understands we studied one testosterone product, and that was the cutaneous one. We didn't study the injectable forms, which seem to have different effects. But when we unblinded the trial, what we found was there was no difference in major cardiovascular adverse events between men that had testosterone and men that didn't. There were some harms. There was an increase in atrial arrhythmias. And we published a second manuscript in the New England Journal of Medicine that there was an increased risk of fractures, which was completely unexpected. We thought that testosterone would improve bone health. It actually made it worse. And
Well,
so there is a concern.
There's an interesting component to that, which one of our other guests talked to us about, and I'm curious what your take is. Is it that the bone mineral density itself went down, leading to more pathologic fractures, or is it that the higher testosterone levels led to riskier behaviors that led to more fractures?
So that latter explanation is an excuse used by advocates for why we found what we found, and I don't buy it for a minute.
Interesting.
We had no evidence that men suddenly became very active and now we're having fractures. So I do think that there is a problem that was unexpected. And, it wasn't a huge problem. It wasn't like people were fracturing, having pathological fractures,
yes.
But there was a vulnerability there that had to be understood.
Understood.
One of the problems is that testosterone treatment has become kind of a cult. And these centers have cropped up all over the country, so-called low T centers. Some of them don't even measure testosterone levels before prescribing testosterone. And if you walk in that door, you're likely to walk out with a testosterone prescription. That wasn't how we used the drug. We used it carefully. And my guess is that when you use it, you also use it carefully.
Well,
and you
we have extremely strict guidelines, as we said. We coach folks through many different aspects of their lifestyle well before we'd ever start that. They need to meet certain guidelines, typically hypogonadism with symptoms. And so this trial also showed a little bit of increased signal in VTE as well, I believe. And
it did.
For us we just need to be careful with these. But like the women's health study, Women's Health Initiative, I think over time there are these lines we sort of learn in medicine, and decades later, sometimes they're proven to be a different.
It happens all the time, doesn't it? Now, just one cautionary note. There appear to be different problems with the injectable testosterone therapies, and blood pressure increases are now part of the label for those products, and so I think we have to be careful not to extrapolate what we found to every potential testosterone treatment. That's just something for people to be aware of. But we didn't, we didn't find anything that really made me overly concerned about, men who need the therapy
Right.
being given the therapy.
Got it. Well, thank you. I appreciate that. I want to pivot and talk about some of your earlier landmark work with what you sort of mentioned with this cardiovascular outcome trial. Some of your early work is really what led to the FDA mandating that CVOT trials are now part of drug approvals. You led some of the initial work with Vioxx, and then again with rosiglitazone, which essentially led to the banning of both of those drugs, led to a very significant improvement, in my opinion, on the recommendations for CVOT as part of these drug approvals. So could you walk the audience through both the Vioxx and the Avandia stories? I think they're fascinating, and I think
they are.
It's going
Well,
to uncover a lot here.
The Vioxx one was an eye opener for me, and what happened was I got put on the FDA's cardiorenal advisory panel in about the year 2000. And the very first panel that I attended was one that was actually put on by the GI division because there was a request to label rofecoxib, known by the brand name of Vioxx, as being safer for the stomach. And when reviewing those materials, and if you've ever seen these advisory committee materials, they're like 500 pages. And if you went deep inside that file, what you saw were the cardiovascular events. And I looked at them, and there were something like 25 MIs. I don't remember the exact numbers in the rofecoxib group, and only 5 in the comparator group, which was naproxen. And it was statistically significant. And it really concerned me. And
the initial signal you saw was not from patient cases in your clinic. It was from this data that you were looking at through the FDA.
That's right. But something actually had happened that made it possible. And what had happened was Public Citizen sued the FDA because until about that year, the materials used to approve drugs were considered confidential and were not in the public domain. Unbelievable that would happen. But Public Citizen won that lawsuit, and the FDA had to start making these files available to people. And so what I did was I went back and talked with some colleagues at the Cleveland Clinic, and I said, look, there's a signal here that something is wrong. And fortunately, Kathy DeAngelis, the editor of JAMA at the time, we approached her, and she was a very bold woman. And she said, look, I know it's very speculative, but we'll take that manuscript. And they did. And we published it, which showed all the cardiovascular.
Were other journals hesitant to take the manuscript because of the implications of what you were saying in that?
We didn't try. We went to JAMA because we thought JAMA was the right place to go. High impact journal. And we thought it would, read by physicians all over the country. And we thought it would allow people at least to be warned that there's a possible signal there that needs to be investigated. And, thanks to Public Citizen, we were allowed to do that. I don't know that anybody had ever done that before, taken data, because it wasn't in the public domain until
and now
very
it is
recent.
mandated that all of this data is in the public domain.
Yeah, they can put some materials, in a redacted form if it's highly proprietary, but it's not done very often. And so fundamentally, it's all out there. And you can do the analysis, and we did it, and we published it. And, we said it was speculative, and we took a terrible beating. I mean, I can't even tell you the things that got said about us by the company that made
So
that drug.
were there actual personal character attacks on you, Dr. Nissen?
100%. I mean, really bad.
Were you ever worried for your reputation or even worse, your life?
Yes. I had a meeting with some folks on Capitol Hill. There were threats. There were actually threats sent. And I had a meeting with people on Capitol Hill. There was a letter being sent around that was extremely nasty, accusing me of being paid off by their competitors. I mean, this sort of thing, but it was sent anonymously. It was just really bad. But what happened was, they actually investigated, and there was a group in Congress, I won't mention who I work with, that they went to the post office and they traced the letters back, and they'd all been posted at a post office that was, within a mile or two of the headquarters of the company that made Vioxx. And so it was pretty clear what was going on. The congressional people were very supportive, and they called up the company and they said, this has got to stop. And eventually it did. But look,
You stood your ground on the data.
Yeah. This is not about me, and I never wanted it to be about me. I always wanted it to be about the science. And so you asked the question, so I gave you an honest answer. But frankly, I never want any of this to ever be about me. It's about patients, and it's about doing the right thing.
Well,
And
look, I appreciate that view, and I think your philosophy there has changed the landscape of the industry, and you should be proud of that, and we have a lot to owe you for that. So thank you for that. I know it's not about you, but
by the way, the sales of the drug dipped a little bit, and then they rebounded very quickly. It went on. It was very successful, multi-billion-dollar drug. It took 4 years before another trial.
Vioxx.
Yeah.
Then 4 years later, another interesting.
Yeah. What happened was they were doing a trial in people with colon polyps to see if it could prevent, I think it was to prevent colon polyps, which are precursors to colon cancer. And in that trial, they were required to monitor cardiovascular events. And their Data Safety and Monitoring Board recommended the trial be stopped because of cardiovascular harm. And it was an explosive result, and it led to the drug being withdrawn by its maker from the market. But it took 4 years. And in that 4 years, we had to weather the storm in order for this to actually happen.
Do you think there are other drugs on the market right now who have harm like that where we just haven't caught the signal yet?
Things have changed. And I'll tell you how they changed. And I did work with the Congress on this. We got clinicaltrials.gov established, and we mandated that you have to post the results of trials or publish them when you do these kind of phase 3 and phase 4 clinical trials. It's not a perfect law, but it creates transparency. And that transparency is very helpful. And I think some of these problems that occurred during this period of time have led to there being a different standard applied. And I think now I'm not so worried about, results being hidden. I'm sure there are some things that just don't make it into the public domain, but I think for the big things, we've made a lot of progress toward more transparency in drug development, which is a good thing.
So you are overall pleased with the FDA's response and the infrastructure we now have in place for regulatory approval?
Well, the FDA tends to be like a lot of bureaucratic organizations, a little bit slow, somewhat defensive. And this came up with the next event that occurred, which was a drug, rosiglitazone, known as Avandia, where FDA was not receptive to our criticism of the drug. And so, but I think there's been
Do you
progress.
think
And
the FDA has an opinion other than science and the truth? I mean, obviously we all understand why the manufacturer would, but wouldn't the FDA be wanting to protect the public?
well, in general, they do. And I'm going to tell you that the FDA is the best regulatory agency in the world. But you have to be careful. And I made one big mistake on the next uh, drug safety issue that came up. What I should have done when this whole rosiglitazone thing emerged was to go to the FDA first
Hmm.
and give them a heads up.
Hmm.
Because when they get blindsided, there's a tendency, and this is true for all of us. If you get blindsided with criticism, you tend to react defensively. And I unfortunately made a mistake and set up that condition where they reacted defensively. And just to tell the story very briefly, I became very concerned about the drug rosiglitazone Avandia. In those days, you could get a drug approved for diabetes if you did a couple thousand patients and showed that it lowered blood sugar. And I was of the opinion that approving drugs because of their biochemical effects rather than their effects on the true health outcomes that matter for patients was not a good idea. We call these surrogate endpoints or surrogate outcomes. And I had been very concerned that that was the wrong thing to do. And what actually happened was there were a couple trials published, one in the Lancet called the Adopt or the Dream trial, and one in the New England Journal called the Adopt trial. And they both showed, favorable effects on glycemic measures with rosiglitazone, but the cardiovascular events trended in the wrong direction, not statistically significant. And so I became concerned and I searched for other trials. And it turns out, another serendipitous event, the now disgraced Attorney General of the state of New York, Elliot Spitzer, had sued the maker of rosiglitazone because they had failed to disclose an increase in risk of suicidality with one of their antidepressant drugs in adolescents. And the settlement for $1 required them to disclose the results of all their clinical trials, which they did on the website. The website was hard to find.
Sure.
But I did find it.
Good.
And when I found it, there were 42 trials that had been conducted with rosiglitazone, 35 of which were unpublished. And we downloaded all the data, and my lead statistician, who is really outstanding, Kathy Wolski, and I quickly analyzed the data. And within just a few days, we sent to the New England Journal of Medicine on May the 1st, 2007, a manuscript describing our results, which showed about a 40% increased risk of myocardial infarction, statistically significant, and cardiovascular death, borderline significant. And the New England Journal of Medicine published that manuscript 21 days later. It was the headline news on in the front page of the Wall Street Journal. It was everywhere. And there was a huge to-do about it. And if the criticism was bad for Vioxx, it was even worse for
Really?
rosiglitazone.
Why do you
Much
think that
more.
is? Because
Well,
it's so much more damning.
yeah, it really was. I mean, it turns out the company had done their own analysis internally and had found the same thing that we found. And so it was a failure to
Do
disclose
you
what was
feel
known.
confident that, just to the last question as well, that kind of stuff doesn't happen anymore? Or do you feel that pharmaceutical companies still find data that they put in the closet?
Look, I think things have improved a lot. I work very closely with the pharmaceutical industry on drug development, and the people I work with are like you and me. They care about patients. They're
Yep.
very ethical. We do a lot of work with companies like Eli Lilly. It's been very good to work with. So, I think things are much better. Are they perfect? Probably not. So are there things out there we don't know about? probably. But I think, we're in better shape. But what happened was, in the wake of the rosiglitazone controversy, the FDA called a meeting in 2008 to discuss what to do. And they invited me to speak. There were actually 3 speakers, outside speakers. It was the endocrine and metabolism panel, so it was not cardiovascular. So I was going into the den of lions, and the 3 speakers were me, Rob Califf, who went on to become the FDA commissioner many years later, and Rory Holman, who founded the UK PDS trial. And I thought about it, and I went in there with a proposal. And here was the issue. If we required a full cardiovascular outcome trial before approving diabetes drugs, it would slow down innovation a lot. So I proposed something that turned out, I think, was reasonably good idea, was that you had to do a small cardiovascular outcome trial and rule out a upper 95% confidence limits for the hazard ratio of 1.8. You had to show that it wasn't worse than placebo or any other comparator. And then you could go on, and then the drug could be approved if you could rule out that pretty liberal hazard ratio, which took a few thousand patients for a few years, but it wasn't horrible. And then you had to go on and rule out 1.3. And to my utter surprise, I wasn't sure what was going to happen. Rob Califf got up and said, I agree with Steve Nissen. And then Rory Coleman got up and said, yeah, I fundamentally agree as well. And this panel of people who initially had been very hostile to our findings about, rosiglitazone, voted to recommend to the FDA that they accept the proposal. And they did. And so something like December 2008, FDA said, this 2-stage process of ruling out a high level of hazard prior to and a lower level of hazard after approval would be now required. And my last couple of slides in that presentation, I said, if we do outcome trials for diabetes drugs, we might just find drugs that are not harmful, but actually have a favorable effect on cardiovascular outcomes. And I made it very clear that I thought once you do outcome trials, you're going to find surprises. You're going to find good things.
Now we see that with the GLPs and SGLT2s and things like that.
It's exactly what happened. And in fact, when the first SGLT2 trial was completed, the company that did it got on a plane and flew from Europe over to meet with me and said, here's the results. What do you think? And I said, you ought to go for approval for an indication for benefit. And they got that indication. And now we found out that the SGLT2 inhibitors reduce risk of heart failure. They improve they reduce the risk of progression of kidney disease. The GLP-1 agonists have very similar benefits. I mean, there's been an explosion of this. And what was interesting is there was a speaker there who was with the ADA, who basically said publicly that if you do what Dr. Nissen is recommending, we will never see another diabetes drug. We'll never have another diabetes drug. But we fought through all of that noise, got it done, and there was a benefit. Now, the FDA no longer requires. They took away that requirement. It was okay.
DOT?
Yeah, it was okay. And I'll tell you why. You can't get a new diabetes drug to be used by anybody unless you show it has these benefits. And so
the regulation sort of shifted the standard of care or the standard of evidence that now we require as clinicians to actually change our practice.
That's right. The other thing was interesting is people said, well, industry will never work with you again because you did this. And that didn't happen. What is now said by a lot of people in industry is that this requirement that I proposed was essential to the success of these drugs. Would people be using relatively expensive branded diabetes drugs if we didn't know that they were better and beneficial.
I mean, if,
And
like you said, if they just change one biomarker like blood glucose, potentially, but then why, we have sulfonylureas and whatever else, right?
yeah,
Insulin,
yeah,
like it's
yeah.
got to be about the outcome.
So it actually had a favorable effect for industry, and I think doing the right thing, if you're willing to take the heat, doing the right thing, ultimately, you got to have faith that it's going to lead to positive outcomes. And, I hung in there with this. And fortunately, people in industry recognized that working with somebody who had the credibility of having, we say when things work and we say when they don't work, that was a good partnership. And I think it actually was more of a help to my involvement with drug development than a harm. Although, interestingly enough, the maker of rofecoxib Vioxx and the maker of rosiglitazone Avandia have never worked with me on
Ever
this.
again.
And I think they have a long memory. And I wish they would. I wish they would, because if they developed a good drug, we'd study it and we'd tell people it was good if it was. So
Well, one thing I appreciate about you, Dr. Nissen, is you're following the truth. You're following the real science. You're following the real data. Where there is benefit, you're going to use pharmaceuticals and be the biggest advocate for them. Where they cause harm, we will protect our patients, though. And I
And
think that
I've been a really strong advocate, for example, for LDL lowering drugs, because I
absolutely.
believe for a long time, and a lot of my early work was on showing that lower LDLs are better.
Well, let's talk
And
about that, Dr. Nissen.
yeah,
I think that's
yeah,
the
yeah,
next sort of
yeah,
area, and I'd
yeah.
love to chat about your early IVIS work in the reversal trial, but your work with bempedoic acid. Why don't we start with, just for the audience, we have again a highly health literate audience here, but as a cardiologist, how do you describe to patients sort of what's happening with cholesterol and how that causes cardiovascular disease?
Well, what I tell people is that the strongest correlate with whether they're going to develop atherosclerotic cardiovascular disease is the time averaged LDL cholesterol over their lifetime. And it's just, the evidence is very strong. And lower for longer is better. And, we did some of the early studies that began to show that. Others have, gone on and done many other studies. And so I have to tell you that I don't necessarily follow the guidelines. The guidelines are written so that you have to have a risk above a certain percent over the next 10 years to be, qualify or to recommend treatment. I look at lifetime risk. If somebody comes into my office and they have an LDL cholesterol of 170 and they're 30 years old, you can't even calculate their 10-year risk because the equations only start at age 40. But if we leave them until they qualify at age 50 for treatment, they've gotten 20 years of having a high LDL. Now, how do we get their time average level down? We got to treat them much more aggressively. And so I've been an advocate for many years of, if you come in and your LDL is 170, I can give you 5 milligrams of a generic statin, or rosuvastatin. I can lower your LDL by at least 40%, and I can do that for years and reduce your time averaged value, and with almost no adverse effects. So, in fact, I've actually been working to take the 5 milligram dose of rosuvastatin into a non-prescription status with FDA and with the maker of the drug. So, lower for longer is better.
Over the counter, just like aspirin, people should be able
Well,
to.
not quite over the counter,
Okay.
but it's kind of a behind the counter where you have to go on into the web and you have to do a web app and put in all your data, and it then calculates if you're a suitable candidate and have no contraindications. So it's a little more complicated, but the bottom line is these drugs are very safe, and I use them earlier. I use them before people's risk over the next 10 years becomes high enough. to qualify based upon the guidelines. And I think that that's the right thing to do.
We
And
certainly
I bet you do the same thing.
we certainly take the same approach, which is we don't believe that risk comes in 10-year chunks, right? We have a lot of patients between 30 and 60, and some of them have risk factors, some of them don't, some of them are on the calculator, some of them are not. The newer calculators, I think, at least will project out what 20 and 30-year risk looks like, but they don't even start at least, until you're 40. So how do you understand what someone's risk is? Well, we triangulate on that through all sorts of different data points. Curious to hear your take on, but your conviction around lower for longer is better is driven, I'm guessing, a little bit by your work with the reversal trial, which actually showed a reversal and regression of atherosclerotic plaque volume with the use of statins. Is that correct?
Not exactly, but let me just tell you what happened. Late 1990s, atorvastatin was coming on the market, and one of the people that was leading that program had followed my work with intravascular ultrasound, a way to really look at plaques inside coronary arteries. And we learned very quickly that once you have the first narrowing in your coronary, practically the entire coronary arterial tree has plaques. And so there was a discussion, and we designed a trial to compare intensive LDL lowering with 80 milligrams of atorvastatin versus a more moderate regimen with 40 milligrams of a less potent statin, pravastatin. And we enrolled about 600 patients. We measured their plaques at baseline carefully, and we developed the technology to do that. I had a team of people that work with me on this. And then we did it again 18 months later. And what we found was there was highly statistically significant progression in the group that got the moderate statin. They had an LDL of around 100, which at the time was considered reasonable. And then the more intensively treated group had an LDL of about 80. And the group that had an LDL of about 80 had no progression. They were essentially no different from zero. So it halted their progression. We then went on later to do a trial called Asteroid, where we used 40 milligrams of rosuvastatin, which was even a more active statin, and we got LDLs down to 60. And when LDLs were 60, about 2/3 of the patients over a 2-year period had less plaque. They regressed. And so we came to the conclusion that getting LDLs down to around 60 was associated with regression. We then went on later, much later, to do a study with a PCSK9 inhibitor, the injectable cholesterol-lowering drugs, and we got LDL down to 36 milligrams per deciliter, and there was even more regression. And so for me, there's just this chain of evidence that suggests that every time we went to a lower LDL level, we got benefits on plaque. And similar studies were done in parallel by others. The Prove It trial studied atorvastatin versus pravastatin, showed benefit on clinical outcomes. And, other trials showed very similar things. Jupiter studied rosuvastatin and showed, benefits on outcomes. And Fourier studied the PCSK9 inhibitors and showed similar.
Absolutely. And, when we talk about sort of regression of the plaques, or at least no progression of that, the logical outcome from that is lower MACE ultimately. Are these trials also actually showing lower cardiovascular events?
So our trials with intravascular ultrasound were too small. They weren't outcome trials. But others did outcome trials like Fourier and like, Jupiter and like Prove It that did study more intensive versus less intensive therapy and showed the same benefits. And so, in our view, what we saw with these mechanistic trials looking at plaques ultimately presaged what was going to be shown in generally later cardiovascular outcome trials.
What is your thought on our sort of targets here on high intensity statin versus an actual LDL goal, like some of the European guidelines have? It sounds like you actually target a number on LDL rather than just having someone on a high intensity.
So I caused more trouble in 2013. I think it was John Lewis that talked about good trouble. And I got in some good trouble in 2013 when the ACCHI guidelines came out and took away targets and said, just give I called it the fire and forget strategy. Give a statin and don't worry so much about what the LDL is. I thought it was wrong. And I went public with it. It ended up on the front page of the New York Times. Both Paul Ridker and I spoke out about it. And, somebody we know very well at the New York Times, a very good science reporter, said, look, there are cardiologists who think these guidelines are wrong and that taking away the targets wasn't a good idea. And that caused, again, I took a beating, particularly from the American Heart Association. They were furious because they're fundamentally a fundraising organization. And anytime they take criticism, they think it's going to affect their fundraising. I had to weather the storm again. And I kept saying to everybody who would listen that taking away targets was not a good idea. And sure enough, eventually the targets did come back. And now they're very aggressive targets in the European guidelines and others, recommending for some people to get below 55 milligrams per deciliter if they're very high risk. And those newer guidelines are right. But it took some people pushing, and we were amongst the people that pushed. I wasn't alone in this, but I certainly started the ball rolling in terms of saying that was not a good idea to take away targets. Patients like targets. They like us to tell them, look, we want you to get somewhere. Yeah.
Heuristics are helpful, and I think even for clinicians and for tracking on population outcomes, things like that. I've heard you talk a little bit about the role of inflammation in the cardiovascular cascade as well. I think this is a divisive topic, and there's this colchicine stuff that came out recently, and people are always wondering, is inflammation sort of just part of the process? Is it riding shotgun? Is it the primary driver of it? We have folks across the spectrum in our practice. Some people are very resistant to statins and the cholesterol theory even, and others are on the other side of that spectrum. And so when we're talking to some patients in that former group, how do you think about that when we're trying to control for inflammation and all these other risk factors?
Yeah. Okay. Let me say a couple of things. First of all, I wrote an editorial in the Annals of Internal Medicine a few years ago entitled Denial, an Internet Cult with Deadly Consequences. And, this statin denial business, you go out on the internet and I did a search with a search engine, and you find more hits for statin harms than you do for statin benefits. And every wacko out there, with the dietary supplements trashing statins, it drives us completely crazy because these drugs have been studied better than any other drugs in history. They're taken by 36 million Americans. And when I see patients that are resistant to statins, it just, it just lights me up.
That's developed? Why do you think statins in particular have been this class of medications that have become demonized?
Well, I just wrote a commentary in the American Journal of Preventive Cardiology that walks through how statin denial came to be. And it's a much longer conversation, but you may want to read it, and some of your listeners may want to read it because it's, again,
notes here.
It's really, really candid. And it doesn't leave anybody spared from criticism for how this happened. But it's like every other kind of fake news, like the anti-vaxxers, everything, all that fake news out there takes hold in the internet and leads people to be resistant. But to turn back to the inflammation story. So when I finished the reversal trial, I wanted to understand why these people did better. And so I looked not just at LDL, but at CRP. And we found that there was a strong correlation between the CRP reduction and the degree of progression, just like LDL. And so we sent that manuscript to the New England Journal of Medicine, and they published it in January of 2005. And interestingly enough, Paul Ridker had done a similar analysis with the Prove It trial, and they took their two manuscripts and they put them back to back in the New England Journal. And then we basically said, we think inflammation is important. It took a long time to prove it, but now it's happened. There was a trial called CANTOS that studied a drug called canakinumab, which was an anti-inflammatory drug that had no effect on cholesterol, and it showed benefit. And then two trials showed benefit with colchicine. Pardon?
Benefit in MACE and reduction in
reducing major adverse cardiovascular events. And then two trials with colchicine, another anti-inflammatory, doesn't affect lipids, showed benefits. So I think that inflammation is important. LDL is important.
It's a two-headed beast. You need to control both. You need to address.
That's right. That's right.
Understood. Do you believe, a lot of your work with bempedoic acid, I think, was revolutionary as well. I'm always surprised that given the statin, pushback and denialism and people that believe they have side effects to it, that we don't see more bempedoic acid. I frankly have maybe only seen it once or twice in my career that anyone's even on that.
Yeah.
But could you tell the audience a little bit about what bempedoic acid even is?
Well, first of all, it's an alternative to statins. It's not as effective. It lowers LDL about 22%, about half what you can get with a good statin. But it also lowers CRP, which is beneficial. And the company that makes it is a small company in Ann Arbor, Michigan. Don't have the resources for marketing that the bigger pharma companies have. So that's part of the problem. And it is proprietary. It's branded, so it's not like the inexpensive generic statins, and that's a barrier. But what we did in a global trial of 14,000 people who were statin intolerant, we made them sign a statement that they couldn't tolerate statins, that they wouldn't take statins, even though they knew statins could benefit them from morbidity and mortality. So we did an ethical trial here in people that just wouldn't take statins. And we gave them bempedoic acid, and it works upstream of where the statins work, but it doesn't get into muscle. So it doesn't cause the muscle side effects that some people do, in fact, truly observe with statins. I'm convinced that some of that is real, some of it is not.
On the order of 5% or so of all.
Yeah, that's a good estimate. Some people say more, maybe 5 to 10, but it's a number. And I see these patients, they get referred to me, and I just saw somebody with a horrible family history and an LDL of 175 and just can't tolerate a statin. And in those people, bempedoic acid can be beneficial, and it can be combined with a cholesterol absorption inhibitor, ezetimibe. If you combine bempedoic acid with ezetimibe, you can get about a 35 to 40% LDL reduction, which is pretty good.
Order of a statin almost, right?
On the order of a moderate intensity statin. And so for a lot of people, that's an alternative. But what we showed was, given over a period of time, that bempedoic acid did in fact reduce the risk of major adverse cardiovascular events. So it's an alternative to statins. It is not as widely used probably as it should be, but part of it is it has cost associated, and part of it is that it doesn't have the marketing clout of a big pharma company, which, frankly, I don't object to that marketing, because when drugs that are beneficial are marketed effectively, it means more people get treated.
Fair.
That's a good thing. That's a good thing.
Fair. Is your practice now to use combination therapy starting out? I know that's a lot of the guidelines, is to start someone initially on a statin and ezetimibe or potentially bempedoic acid as well.
I do it all the time. I do, and, the idea is you can calculate based upon the known typical effects of these drugs, what the likely LDL is going to be with various approaches. If your LDL is 180, then you're probably going to need combination, and your goal is 70. You're probably going to need more than one drug to get there. And so you might as well just start out with a reasonable
Lower down
therapy.
on both,
Yeah.
right? Reduce
Yeah.
side effects. So
Yeah.
let me pose a question on a patient case, because this kind of patient comes to us often. They are relatively metabolically healthy, good blood pressure, good weight, elevated ApoB 120 to 150, low A1C, no major family history of ASCVD, low lipoprotein A, and low CAC, 50 years old.
Yeah.
Would you treat that patient? They have an isolated ApoB and nothing else. I think that's the
Well,
question that
yeah.
people try to
Yeah.
try.
Well, their ApoB is up, but if their ApoB is up, their LDL is also up.
Fair. That's what I
So
mean.
the LDL, yeah. So the ApoB is 120. The LDL is probably going to be 140. Yeah. Yeah. I think this is a great opportunity for shared decision making. And I'll bet you anything that's what you do. I sit down with the patient and I say, look, this is what we know. We know that your average lifetime LDL is a pretty good predictor of whether you're going to get disease. You are low risk. You don't have a family history. You don't have diabetes. You don't have hypertension. I'm assuming this person is really pretty healthy.
Yeah. Yeah.
And I say to them, it's not clearly been shown that you will benefit, but you might. And we could treat you with ezetimibe, a drug that's incredibly well tolerated, really cheap. Or it's going to lower your LDL 18, 20%. It'd be a pretty cheap insurance policy against later developing disease. We could treat you with a low dose of a statin. I typically use 5 milligrams of rosuvastatin.
Me too.
Or we could watch and wait and see what happens. And we have a discussion. And I typically take an hour with every new patient.
Good.
And, we go over what we know and what we don't know. And we make a decision together. And that's the right way
Thank you
to
for
go
that.
about
I
it.
mean, I agree. That's exactly how we approach it. Our appointments are 90 minutes. And, I've been a PCP. My dad was a family doc for 40 years. I feel the pressure and I feel for my PCP colleagues who have 7 minutes with their patients trying to have these complex decisions on, beliefs and what the evidence shows and what our alternatives are. And so I appreciate that you're able to have that level of discussion. I think one thing that we like to do is really give ourselves as much data as possible. Two pieces of data that we really believe that most patients should have, depending on their age and risk factors, lipoprotein A and coronary calcium scoring.
I agree with you. But one thing we don't do is do like NMR lipid profiles.
Tell me a little bit about that, because I've heard you say you're vehemently against sort of the particle count. We like the insulin resistance scores that we sometimes get, but managing towards particle counts, I believe, have not been actually studied. Is that the criticism?
They have not been studied. I think we get everything we need from
Standard panel.
ApoB, LDL, and non-HDL. And, you have all those things, and triglycerides, and you have all those things. You're going to really have very solid evidence, and all of them have solid evidence for, their association with risk. Now, we haven't been able to show that lowering triglycerides can reduce major adverse cardiovascular events, but I think it's a pretty good target. And you're right about lipoprotein A. We've got a lot of studies now ongoing. It is a very powerful risk factor. About 20% of the population have. Most of them don't know that they have it. I did a study in 48,000 people in almost every country in the world that had ASCVD. And only 13.9% of them had their lipoprotein A checked. You're way ahead of the curve because you know that it's something you have to do routinely. And it's a huge risk amplifier. And now you take that patient with an ApoB of 120 and give them a lipoprotein A of 175 nanomoles per liter. And I'm going to treat that patient every time. I'm going to recommend treating them. Now, they have to make the decision together, but it's the right thing to do.
That's how we sort of see it too, is lipoprotein A. And I want to go deeper, much deeper on this. As a genetic risk factor is often taught to be checked once. To our understanding, it is part of the ApoB family. Is that right? But it has an ApoA protein on it as well.
That's right. That's correct.
And lipoprotein A, particle for particle, I've seen studies show that it is something like 6 times more atherogenic than an LDL particle. Is that right?
It is certainly, it's debatable, but somewhere between 3 and 6 is a reasonable, guess. These are hard studies to do.
Sure,
and so,
I understand.
But the bottom line is it's very atherogenic and by the way, prothrombotic.
Absolutely.
Which is another aspect of it that makes it a particularly noxious particle.
We think about lipoprotein A as a genetic marker. The guidance is often to check it once and you're sort of done. What is your experience with that? And I'd like to pose a question to you based on our anecdotes as well.
Well, first of all, it does vary from blood lab, and I don't understand all the reasons why. Some of it really is laboratory to laboratory. But I wished it were perfectly stable, but it's not always. And so we've seen people that come in, we think they're qualified for a clinical trial, which requires 175 nanomoles or greater, and they're at 200. And then when we repeat a baseline for consideration of the trial, it's 130. And I'll bet anything you see that too.
I do, because sometimes I get in, it's elevated, and like most things, I want to double check, right? I want to make sure that this thing is real. Let's make sure there wasn't some lab error. Honestly, Dr. Nissen, probably 50% of the time I recheck a lipoprotein A, it is vastly different than the first time I checked it.
Yeah,
and so
it's certainly, I wished I understood that. I really wished I understood it. It is a problem.
You think that's a lab assay issue, though, and not that the physiology of lipoprotein A is actually changing that dramatically in someone?
Well, I do think it does change. I mean, it changes around menopause. There are other things that have an effect on lipoprotein A. I just don't think we have as good a lab assays as we need for this. And it doesn't have perfect stability. Now, you're going to see the same thing with CRP and with LDL. And so it's probably in the same ballpark, in terms of the variability. I've also seen people that come in to get an LDL and you repeat it and it's somewhat different. Triglycerides are even worse. If you have a bowl of ice cream last night, your triglycerides are going to be up. We have to make decisions under conditions of some level of uncertainty. But in general, if somebody comes in and they're 300 nanomoles per liter, they might come back. They got a problem. They may come back at 225.
Doesn't matter. You're still at a high.
Doesn't matter.
Right.
It's still high.
It's directional more than anything.
That's right.
Why do you think more people don't check lipoprotein A? It's in the guidelines. I mean, certainly in the European guidelines, it's meant to check. I mean, to me, it seems like something every PCP should know about, but very few patients have ever had that check.
So I'm going to make some people angry with me, but I'm going to say it anyway because this is who I am. There's a problem sometimes called clinical inertia. And, people have always done things one way and they keep doing that the same way. Clinical inertia with primary care physicians is a huge problem with LDL cholesterol. I see notes from primary care physicians. Somebody comes in, they got a lot of risk factors and their LDL is high. And the note said, counsel patient on improving their diet. And they come back in 3 months or 6 months, it's still high. And it still says, advise the patient to eat less saturated fat, whatever. And they don't pull the trigger on treatment. And it is a problem. And I think, there's nothing better in medicine than preventive care. I worked for most of my career in a coronary care unit, and seeing a 45-year-old man coming in with a big anterior wall MI is something I never want to see again. And so we've got to get people to be more aware, and we're doing everything we can educationally, but it just doesn't trickle down.
Even conversations like this are helpful, but I agree. I mean, there's all sorts of studies that talk about, guideline-driven therapies that are endorsed by professional society still often take a decade or more to diffuse into clinical practice.
They do. Yeah. By the way, our fellows like to use a term that I absolutely abhor, GDMT. Guideline-directed medical therapy. And let me say this as clearly as I can. Guidelines don't direct medical therapy. Physicians direct medical therapy. And guidelines are guidelines. They're not mandates. They're not rules. They're giving you an idea.
I think I very much believe that, and I go off the guidelines all the time because I've had shared decision-making with my patients, and
That's right.
For the the risks and benefits and alternatives. But I feel that in most traditional practices, especially in a malpractice driven defensive medicine environment, the top cover for most doctors is to follow the guidelines, so that if something bad happens, they can say, well, I follow the guidelines. Like, what do you want me to do? Right. If you go off, though, you don't have that protection. And so there's a weird dichotomy, I think, that docs are caught in sometimes, where even though they might understand physiologically that there might be off-label uses or to consider things differently, they're stuck following the guidelines because some administrator is following those numbers.
Okay, so I'll say this as clear as I can, and I know you believe this because I can tell you do. Our job is to be advocates for our patients. And this defensive medicine and all of that, look, I don't always follow the guidelines. I do what I think is right based upon evolving science. And by the way, the guidelines typically lag the evolving science by years, because the guideline committees, meet and they work forever on writing the guidelines, and they're just not even up to date some of the time. And so if you practice, the term I prefer to GDMT is evidence-based medical practice. And where you synthesize the evidence, you present it to the patient. And then the other thing that I think is very protective is I'll write in the chart, I discussed this with the patient, and together we made a decision to do X. Because it is shared decision-making. Exactly. And that's a very protective approach. If you want to protect yourself.
Medicine has gotten away from that in some aspects. There's a patriarchal approach, but at least where we can, we do everything to involve our patients in the decision-making process. I want to ask one question about the LDL and the time under curve, which you mentioned earlier, and then we'll come back to LPA. But I am curious, is there any amount of lifestyle that you think can get someone to an LDL or ApoB goal if I have a strong family history, right? Like my father has heart disease, my first cousin, my grandparents, I've had diabetes, I've been overweight, I'm fine now, but is there any amount of lifestyle I can do to get my ApoB to an appropriate level? And is that appropriate level actually 0? And would the logical conclusion then be that basically everyone should be on some kind of therapy to get their ApoB and LDL down as low as possible?
First of all, look, diet and exercise and all the good things we recommend for patients is a good idea. And actually, exercise is probably as good as almost any drug. If you get people off the couch and get them out exercising, I try to get them to get 10,000 plus steps a day. That's pretty effective. But only about 10 to 15% of LDL cholesterol comes from the diet. That's about the amount you can affect with a drug like ezetimibe. Yeah. And so if you're more than 15% above where you really ought to be, you're not going to likely get there. Now, there are people that have these extreme diets. There's this guy, Esselstyn, here, that has a diet that drives me crazy. He tells people they can't use any oil at all. And I tell people to liberally use olive oil, which, by the way, has very good evidence for benefits. a Mediterranean diet's a good diet, and I tell them that. But it isn't going to lower their LDL cholesterol more than 10 or 15%. And so that's why it's always a good idea. And let me tell you why diet is such a great idea. It's the rule of 6. And you know the rule of 6. If you double the dose of a statin, you get 6% more LDL lowering. So let's take rosuvastatin. If you go from 5 milligram dose to a 40 milligram dose, you get 18% more LDL lowering. If you could get that 18% from diet, you could stay on 5 milligrams of rosuvastatin, which has fewer side effects, a better safety profile in every way. And so diet can amplify the benefits of cholesterol-lowering medication, and it's generally a good idea. And I know you know about the Predimed study, which
Yes.
was a study done in Spain showing that an olive oil-enriched diet, Mediterranean diet, compared with the usual diet, reduced the incidence in primary prevention patients of atherosclerotic vascular events.
Absolutely. Yeah, we start with the core 4 always: diet, exercise, sleep, and mental health and relationships. But ultimately, I am still curious on this very direct question: is if someone has an ApoB or an LDL of 70 and they have high risk factors, like, should they still be on medications? If you believe the target should essentially be 0 for everyone, should those people be on therapy?
Yeah, I haven't gone that far,
Okay.
but I probably have more aggressive and lower targets than most physicians you're ever going to encounter. Because if I have a safe therapy, look, if your ApoB is 70 and you take a very small dose of a statin and you lower it to 50, in a high-risk patient, that probably has benefits. And so, by the way, in some of our trials, we have had patients with LDLs in the single digits.
Wow.
And they're fine.
No negative. Sometimes people will say there's more infection risk or higher dementia risk or things like that, but we haven't seen any of that in the big trials.
There's actually lower dementia risk. Vascular dementia is one of the more common causes of dementia. And you see now Brown and Goldstein, who won the Nobel Prize for discovering the LDL receptor, in their Nobel Prize lecture, they said that they thought as a species that we had evolved, that we don't really need an LDL more than 25 or 30. And they gave some of the evidence for why that's the case. In some primitive populations, that are maybe a little bit undernourished, but, who are exercising constantly, they have LDLs that are really low.
Right.
So low LDL is not a bad thing. I don't know that we have benefits. The lowest we've studied in trials would be Fourier, and they went to an LDL of 30, and they did a big trial on neurocognitive function in those patients called Ebbinghaus, published in the New England Journal of Medicine, that showed there was not a negative neurocognitive effect at an LDL of 30. So I think, if you need to go there, you can do it.
Amen. Okay. Thank you for that. That was very helpful. Coming back to LPA, this is another factor that we continue to talk about, has huge risk here. We don't have things in our lifestyle that we know of that have been studied that can bring this down. I think there's been some stuff around like aspirin and niacin, but not great evidence on any of those things. Tell us a little bit, Dr. Nissen, about some of the ongoing trials right now. I know there's some oral forms, some injectable forms of some of the therapies that are on the horizon for lipoprotein A.
So we've studied several therapies. And because it's a genetic risk factor, the only current way we have to treat it is to interrupt it's a product of a gene. So we have to interrupt the product of the gene. And the way that's done is DNA codes for lipoprotein A. It makes messenger RNA, and messenger RNA instructs cells to make a protein, in this case, apolipoprotein A. So we have the first of these therapies to be developed is what's known as an antisense oligonucleotide, or ASO. And it's a small piece of single-stranded DNA that's complementary to the messenger RNA, but when it finds a messenger RNA, it degrades it. It causes it to be degraded. If you don't make message, you don't make the protein. Yeah. And you don't make the protein, you don't make the particle. And in a phase 2 trial, it lowered lipoprotein A by 80%. And that has been in studies. I helped to design the study back in 2018, 2019. Been going on for a number of years, did it all through the pandemic, which was hard. We're treating patients with 80 milligrams every month. And we're blinded, but it's 8,300 patients, and it's due to be completed sometime next year.
And is the primary outcome a reduction in lipoprotein A biomarker, or is it reduction in cardiovascular outcomes?
Reduction in cardiovascular adverse events. It's a MACE trial. So it's the right answer. And the FDA will not approve these drugs until we show that they can reduce. And the FDA is right. This is the right thing to do.
So in many ways, your earlier work on pushing the FDA that you got to actually show outcome benefit with these drugs is now being applied to these lipoprotein A.
I don't know that our work had any effect on it, but I do think it's the right thing to do for sure. And, and then along have come several other drugs. Amgen developed a drug called olpasiran, which is a small interfering RNA. And Lilly developed another small interfering RNA known as lepodisiran. Olpasiran in phase 2 lowered lipoprotein A more than 90%, and it's in a ongoing phase 3 trial. And lepodisiran, we published this year in New England Journal of Medicine, the 95% reduction in lipoprotein A with this drug. Now, these drugs, these small interfering RNAs, are double-stranded RNA, and they can be given less frequently.
Weekly or monthly?
The Amgen drug is given 4 times a year, every 3 months, and the Lilly drug is given twice a year, every 6 months. So we're getting 95% reduction with a twice-a-year therapy. And actually, in some people, levels were lowered by about 90% time averaged when given once a year, like a vaccine. So, there's been a lot of progress. But look, what it means is we've got a bunch of shots on goal, and we just hope at least one of them ends up in the back of the net. And maybe all of them will. And then finally, there's a drug called muvalaplin, clever drug that prevents...
Oral one?
Pardon?
This is the oral one?
This is the oral one, yes, sir. And it prevents the linking of the apolipoprotein A with the core particle that contains ApoB. And if you don't link the two together, you don't make lipoprotein A. And it lowered, recently published in JAMA phase 2 trial, by about 85%. So it's pretty effective.
These are blockbuster drugs then, effectively. If they, assuming these get one of them makes it through. Yeah. What is your take on sort of like a pragmatic rollout? Would you then recommend that every single patient gets an LPA, and if they are elevated, that they essentially start this medication?
Well, in one of the trials, the trial that we're doing with lepodisiran, we have a mixture of primary and secondary prevention patients. So actually more primary prevention, more of the patients have never had an event than the number that have actually had an event. And they're doing a dedicated primary prevention trial with olpasiran. And so we will find out whether we can prevent a first event in high-risk patients and whether we can prevent a recurrent event in people that have already had an event. And so we're moving along on this, and we don't know what the answer is yet. We're hoping to have some answers soon. And, we'll tell it like it is. And I'm praying that it's going to work, because I know how much suffering is caused by premature cardiovascular disease due to a high lipoprotein A. And I'm trying to get people to test for it. And if any of these drugs is successful and gets approved, we're going to need a global campaign to identify patients who will benefit. Now, please understand that in the primary prevention cohorts, we're not studying low-risk patients. We're studying people that have other risk factors.
Yeah.
You have to start somewhere.
I hear you.
And so we're starting at high risk.
But
if it works,
high risk,
it will do.
I mean, that's going to probably, account for 50% of the adult population in the United States. If high risk includes a comorbidity like hypertension, hyperlipidemia.
Yeah. Well, in the studies that are being done in these people, you have more than one, risk factor. But the point about this is that there are plenty of people out there,
Right.
millions of them, that would potentially qualify, and we want to help them.
I love it. Well, thank you for your work on all of this, Dr. Nissen. In our last couple of minutes, I am just curious to turn it back to you for a moment. And we see a lot of patients who are coming to us because they want to live their best life right now, but they also want to be here 20 and 30 and 40 years from now for their grandkids. I'm curious, what is your sort of next 20 and 30 years look like? I won't out your age here, but are you?
I'm gonna out my age because I'm not, it doesn't bother me. I'm 77 years old, and I'm the first one here in the morning, get here at 4 in the morning. And I work really hard. I like what I do. I care about what I do. I want to help patients, to provide benefits. And I think I'm in a position now to be able to do that. And I'm going to do this for as long as I can. And I like to do this. Gene Bernwald, the most famous cardiologist on the planet, is 95 years old and still writing articles. And Bertram Pitts in his early 90s is still writing articles. And so if you like what you do, and now I exercise regularly. I'm thin. I eat a careful diet. I keep my LDL very low.
You have a personal stack of medications. Do you do any supplements? You take a medication to keep your ApoB?
No, and I advise people not to take supplements. They're unregulated, and there was a law passed in 1993 that was a disaster called DSHEA that bars the FDA from regulating dietary supplements. They're regulated primarily by the Federal Trade Commission. We can't be certain of what all of them contain. They've not been studied in rigorous trials. And I tell patients not to take supplements. Some of them interact with prescription medications. We don't know about those things. And they're not a proven benefit. And so we did a study. My colleague, Dr. Laffin, the next office over, did a study called the SPORT trial. I don't know if you've read it, but we studied 6 dietary supplements that are claimed to benefit cholesterol. And we studied them in comparison to placebo and 5 milligrams of rosuvastatin. And none of the supplements showed any benefit on cholesterol. And one of them, garlic, raised LDL cholesterol. Widely marketed.
Interesting.
So without rigorous trials, I cannot recommend dietary supplements.
Well, I appreciate that view strictly on the evidence here. And I think that's the prudent approach as well. So you have a personal stack with medications to keep your LDL as low as possible. You follow a mostly Mediterranean diet?
Yes, I do. Yep.
You exercise daily, get all your steps and increase your VO2 max. And anything else you do for your personal, like if you want to be 97 and still publishing papers, anything else you do for yourself?
Yeah, I try to get a decent night's sleep, although, it's hard because I work very hard. But, that's it. I mean, diet, exercise, a low LDL. My blood pressure is low, but I do take something for it, and I keep it below 120, which I think is it's a good thing. We didn't talk about blood pressure, but, lower is probably better there too. And, I don't think there's any magic here. And, there is a reason why I'm still able to work really hard at age 77, is I take good care of myself. And I know what you do, and you provide that sort of care to patients, that you help them understand what they have to do to stay healthy and have a lot of longevity and enjoy life. And I enjoy life, and I enjoy what I do.
I can tell, and it's infectious as well. I think having purpose is something we talk about often. And I think that's something that's very clear in your work. Dr. Nissen, thank you for sharing all that you've shared today with our listeners, with me. Thank you for your advocacy. I mean, just hearing your stories and learning more about you, your work has had rippling effects on the public, and we all owe you a debt of gratitude. So thank you. I know it's about the science here, and you've pushed us all to appreciate a better, higher level of science and demand that of everyone around us. So I really appreciate you being here. We will carry these messages forward. And for all of you listening, thank you for spending another episode of The Ultimate Asset with us. We hope you learned as much as I did on this wonderful podcast with Dr. Stephen Nissen, and we will be back next time talking more about heart health. Thank you.
And thank you for having me.
Thank you so much, Dr. Nissen. Have a great day. Thank you for listening to The Ultimate Asset. Until next time, keep investing in your greatest asset, yourself. This podcast is for informational purposes only and does not constitute professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified healthcare provider with any questions you may have regarding a medical condition or before making any changes to your health regimen. Never disregard professional medical advice or delay seeking it because of something you've heard on this podcast. Reliance on any information provided in this podcast is solely at your own discretion.